Today's topic is mediators of inflammation and leukocyte extravasation. Inflammation is a complex response to injury or a trauma. It involves various immune systems and numerous mediators. The mediators in inflammation play a significant role in assembling various cells and in eliciting and regulating the immune response. One of the key mediators of inflammation is the chemokines, a family of cytokines with chemoattractive properties. Other mediators of inflammation are the products of four major systems in the plasma: the kinin system, the clotting system, the fibrinolytic system, and the complement system. Besides the mediators of inflammation, the other important characteristics of inflammation is the assembling of various cells in the inflammatory sites. This assembling of cell is accomplished by controlled migration of leukocyte population across the endothelial layer of blood vessels, termed leukocyte extravasation. Mediators of inflammation, chemokines. Chemokines are the key mediators of inflammation. They are a family of small polypeptides with about 90 to 130 amino acid residues in length. They selectively and specifically control the adhesion and chemotaxis of many types of leukocyte population and subpopulations. They also play a significant role in activation of many immune cells. Some chemokines are primarily involved in inflammatory processes, while others are constitutively expressed and play important role in hemostasis and cellular development. Inflammatory chemokines are typically induced in response to infection. Contact with pathogen or the action of pro-inflammatory cytokines such as TNF-alpha upregulates the expression of inflammatory chemokines at the site of developing inflammation. Chemokines also causes leukocytes to migrate into various tissue sites by inducing the adherence of these cells to the vascular endothelium. After migrating into tissue, leukocytes are attracted towards the sites where there are high concentration of chemokines. This results in targeted recruitment of phagocytes and effector lymphocytes to inflammatory sites. There are more than 50 chemokines identified till date and about 15 chemokine receptors. All chemokines possesses 4 cysteine residues, and depending on the position of 2 of the cysteine residues, they are grouped as CC subgroup chemokines, in which the conserved cysteines are contagious, and CXCR subgroup chemokines, in which the conserved cysteine are separated by some other amino acids. Chemokine action is mediated by binding to receptors. Chemokine receptors have a distinct structure where the polypeptide chain traverses the membrane 7 times. There are 2 subgroups of chemokine receptors: CC receptors or CCRs, which recognize CC chemokines, and CXC receptors or CXCRs, which recognize CXC chemokines. The receptor profile of various immune cells mediates its activities. When a receptor binds to an appropriate chemokine, G protein associated signaling transduction processes are activated. This generates a number of potent second messengers like cyclic AMP, IP3, calcium, and activated small g proteins, which affects gene expressions. Addition of an appropriate chemokine to leukocytes causes abrupt changes within seconds. On addition of chemokines, leukocyte changes in shape extensively promotes greater adhesiveness to endothelial walls by activation of leukocyte integrins and also generates microbicidal oxygen radicals in phagocytes. This activation of signal transduction pathways promote other changes such as the release of granular contents like proteases in neutrophils and macrophages, histamine from basophils and cytotoxic proteins from eosinophils. Other mediators of inflammation. Plasma contains 4 interconnected mediator-producing systems: the kinin system, the clotting system, the fibrinolytic system, and the complement system. The first 3 systems share a common intermediate called the Heckman factor. When tissue damage occurs, these 4 systems are activated to form a web of interacting systems that generate a number of mediators of inflammation. In the kinin system, the Heckman factor activates prekallikrines to form kallikrines, which further cleaves kininogens to produce bradykinin. This inflammatory mediators, bradykinin is a potent basic peptide that increases vascular permeability, causes vasodilation, induces pain, and induces contraction of smooth muscles. Kallikrine also acts directly on the complement system by cleaving C5 into C5a and C5b. The C5a complement component is an anaphylotoxin that induces mast cell degranulation, which results in the release of number of inflammatory mediators from the mast cell. Damage to blood vessel also trigger a cascade of enzyme activities that leads to production of large quantities of thrombin. Thrombin activates the soluble fibrinogen in tissue fluids or plasma to produce insoluble strands of fibrin and fibrinopeptides. The insoluble fibrin strands crisscross one another to form a clot which serves as a barrier to the spread of infection. The fibrinopeptides produced is a potent inflammatory mediator which induces increased vascular permeability and neutrophil chemotaxis. Fibrin clots are further removed from the injured tissue by the fibrinolytic system. In the final step of the fibrinolytic pathway, plasminogen is converted to plasmin, a potent proteolytic enzyme. Plasmin degrades fibrin clot into products which act as chemotactic agents for neutrophils. Plasmin also activates the classical pathway of complement and thus contributes to inflammatory response. Activation of the complement system results in the formation of a number of complement split products that serves as important mediators of inflammation. Binding of the anaphylotoxin C3a, C4a and C5a to receptors on the membrane of tissue mast cells induces degranulation which causes release of many mediators. These mediators induce smooth muscle contraction and increase vascular permeability. C3a, C5a and C5b67 causes migration of monocytes and neutrophils to the injured tissue. Thus, activation of the complement system results in influx of fluid that carry antibody and phagocytic cells to the site of tissue injury. Lipids as inflammatory mediators. Tissue damage also leads to degradation of membrane phospholipids into arachidonic acid and lysoplatelet activating factor. Lysoplatelet activating factor is subsequently converted into platelet activating factor or PAF. PAF causes activation of platelets and it also plays important role in chemotaxis of eosinophils and in the activation and degranulation of neutrophils and eosinophils. Arachidonic acid is further metabolized by the cyclooxygenase pathway which produces prostaglandins and thromboxanes. Different prostaglandins are produced by different cells and they have diverse physiological effects including increased vascular permeability, increased vascular dilation and induction of neutrophil chemotaxis. Thromboxanes are involved in platelet aggregation and also causes constriction of blood vessels. Arachidonic acid is also metabolized by the lipooxygenase pathway to produce leukotrienes. Leukotrienes mediate smooth muscle contraction and neutrophil chemotaxis. Cytokines as inflammatory mediators. Besides the chemokines, many of the cytokines also play important role in inflammatory response. Cytokines like interleukin-1, interleukin-6, TNF-alpha and interleukin-12 exhibit redundant and pleiotropic effects that together contribute to the inflammatory response. They are involved in various inflammatory functions like induction of acute phase proteins, increased vascular permeability, increased expression of adhesion molecules on endothelial cells, platelet production, chemokine induction, T cell and B cell induction, etc. Interleukin-12 is also an inducer of T helper 1 differentiation which is important in chronic inflammation. Interferon gamma also plays significant role in chronic inflammation by attracting and activating macrophages. Cell adhesion molecules. The vascular endothelium plays an important role in monitoring and regulating the movement of blood-borne molecules and leukocytes into the tissues. Circulating leukocytes enter the inflamed tissue or peripheral lymphoid organs by adhering to and passing between the endothelial cells lining the wall of blood vessels by a process called extravasation. Endothelial cells therefore express leukocyte-specific cell adhesion molecules or CAMs to facilitate extravasation. Some of these membrane proteins are expressed constitutively and others are expressed only in response to local concentration of cytokines produced during an inflammatory response. Circulating leukocytes bear receptors that bind to CAMs on the endothelial cells that enable them to extravasate into tissues. CAMs on leukocyte also serve to increase the strength of the functional interaction between cells of the immune system. There are four major families of CAMs. They are selectin family, the mucin-like family, the integrin family, and the immunoglobulin superfamily. The selectin family of membrane glycoproteins has a distal lectin-like domain which binds with carbohydrate moieties. Selectin interacts primarily with ciliated carbohydrate moieties present on mucin-like molecules. There are three types of selectin molecules designated as L, E, and P. Most circulating leukocytes express L-selectin, whereas E-selectin and P-selectin are expressed on vascular endothelial cells. Selectin molecules are responsible for the initial binding of the leukocytes to vascular endothelium. The mucins are a group of serine and threonine-rich proteins that are heavily glycosylated. Their extended structure allows them to present ciliated carbohydrate ligands to selectins. For example, L-lectin on leukocytes recognizes ciliated carbohydrates on two mucin-like molecules CD34 and glycam-1 expressed on certain endothelial cells of the lymph nodes. The integrins are heterodimeric proteins consisting of an alpha and beta chain that are expressed by leukocytes and facilitate adherence to vascular endothelium. Different integrins are expressed by different populations of leukocytes, allowing these cells to bind to different CAMs that belong to the immunoglobulin superfamily expressed along the vascular endothelium. Some integrins must be activated before they can bind with high affinity to their ligands. Many adhesion molecules also contain a variable number of immunoglobulin-like domains, and they are classified in the immunoglobulin superfamily. Some of them are ICAM1, ICAM2, ICAM3 and VCAM, which are expressed on vascular endothelial cells and bind to various integrin molecules. Leukocyte extravasation. Leukocyte extravasation is the adherence and passing of the leukocyte across the vascular endothelium layer to reach the inflamed site. The first cells to undergo extravasation during inflammation are the neutrophils. The process of neutrophil extravasation can be divided into four sequential steps: rolling, activation by chemoattractant stimulus, arrest and adhesion, and transendothelial migration. In the first step, neutrophils attach loosely to the endothelium by a low affinity selectin-mucin interaction. This interaction attaches the neutrophil briefly to the endothelial cells, but the shear force of the circulating blood soon detaches the neutrophil. Selectin molecules on another endothelial cell again tether the neutrophils, and this process is repeated so that the neutrophil tumbles over along the endothelium, showing a rolling action. As the neutrophil rolls, it is activated by various chemoattractants. Some of the chemoattractants are interleukin-8, macrophage inflammatory proteins, platelet activating factor, the complement split products like C5a, C3a, and C5b67, and various N-formyl peptides produced by the breakdown of bacterial proteins during an infection. Binding of these chemoattractants to receptors on the neutrophil membrane triggers an activating signal mediated by G protein associated with the receptors. This signal induces a conformational change in the integrin molecule in the neutrophil membrane, increasing the affinity for the Ig superfamily adhesion molecules on the endothelium. This enables the cell to adhere firmly to the endothelial cells. Subsequently, the neutrophil migrate through the basal walls into the tissue. Various subsets of lymphocytes also exhibit directed extravasation at the inflammatory site and secondary lymphoid organs. The overall process of extravasation is similar to that of the neutrophils, involving the four major steps. Some regions of vascular endothelium in post-capillary venules of various lymphoid organs are composed of specialized cells with a plumb cuboidal shapes. Such regions are called high endothelial venules or HEVs. They are the major sites where leukocyte extravasation takes place. HEV express a variety of CAMs that facilitates extravasation. In conclusion, the complex immune response in inflammation is possible with the help of the varied mediators. One of the key mediators is the chemokines, which attract and aids in the recruitment of various cells in the inflammatory sites. Besides the chemokines, there are other mediators that facilitate extravasation of cells across the vascular endothelium by causing vasodilation and increasing the permeability of the vasculature. Cell adhesion molecules also play a significant role in attachment of the cells and facilitates extravasation of leukocytes during inflammatory response. Recruitment of various cells in the inflammatory site is a hallmark of inflammatory response and is essential to have a combined and coordinated response against invading pathogens.